The third lecture of this session is given by Jing Yu, and the title is Structure of Polyelectrolyte Brushes in the Presence of Multivalent Counterions.
Thanks for the introduction, and good afternoon, everyone. Today I would like to talk about the effect of multivalent counterions to the structure of polyelectrolyte brushes. The majority of the story actually was done at University of Chicago, where I did my postdoc, and many thanks to my postdoc advisor, Matt Terrell, as well as my collaborators, Wanda Pavlo and Marina Ruth. And a lot of work was done by Nick Jackson, who did most of the simulation. And also Jing Yu, who's now my postdoc working on AFM. Polyelectrolyte brushes are very important in many colloidal applications. They can make colloidal systems stable. Also, they're very useful in drug delivery systems like the DNA brushes. We're really interested in polyelectrolyte brushes, just as Nanjun said. They're really good lubricants in many lubrication systems in our human body, such as in the joints. Knee joints or hip joints, you have charged polyelectrolytes, such as hyaluronic acids, as well as glycosylated proteins, form these brush-like structures, which play a very important role in the boundary lubrication properties of the knee joints, and therefore we're really interested in the brushes. Now, to understand the properties of the brushes, it's very important to understand the structures of the brushes. The brush system really relies on this extended brush layer to provide a lot of its functions. For example, provides repulsions between surfaces and make the colloidal system stable. And in the solution, because the polyelectrolyte chains are charged, so the solution ionic environment will dramatically affect the structure of polyelectrolytes. One example is the multivalent counterions. Now, when I first started this project, we already had some evidence showing that the presence of multivalent counterions, trivalent counterions in this particular case, even at very low concentration, at a concentration about 1 in 1,000 ions in the solution, can dramatically alter the surface forces between two phase-resolved PSS brushes on surfaces, as the surface force apparatus measurement shows that the interaction between two PSS brushes at low graphene density is purely repulsive indeed in monovalent salt solution. But when we add trivalent ions, either lanthanum or this ruthenium hexaamine, we dramatically reduce the repulsion force. Also, in some situations, one indeed can measure strong adhesion force between two polyelectrolyte brushes, and we believe this is due to electrostatic cross-linking mediated by the trivalent ions. Also, the surface force measurement again shows the height of the brush, which is very important to the functionality of the brushes, dramatically decreases when you have about 1 in 1,000 ions in the solution replaced with multivalent ions. Now, understanding the effect of multivalent ions is extremely important. With the advances of surface-initiated polymerization, I started making dense polyelectrolyte brushes. Previously, we can only make PSS brushes through surface absorption. But I'm really interested in what happens at high grafting density. So with ATRP, we can graft the PSS brushes on microsurface with a fairly high grafting density, close to 0.1 chain per square nanometer. And when we measure the force distance profiles with SFA, which measures the interactions between two PSS brushes, we see that again in monovalent salt, the force are purely repulsive. The force is purely repulsive and fairly reversible. But when we started to add a tiny bit of trivalent etron ions in the solution, we started to notice the range of repulsion dramatically decreases, and if we further increase the concentration to 0.1 millimolar or 0.5 millimolar, we see the range of repulsion decreased by about 70%, and the structure sort of stayed there, doesn't further change after the PSS brushes fully collapses. Now, based on that, we believe the trivalent ions will replace the monovalent counterions in the solution, which causes the collapse of the brushes, as just Professor Matthias talked about on Monday. Now, this collapse of the PSS brushes is indeed reversible. After you collapse the brushes with multivalent ions, if you remove the multivalent counterions in the solution and replace the solution with monovalent ions, the brush start to reswell. And you can accelerate this process by using a high concentration of monovalent salt solution, which will replace most of the trivalent counterions out. After that, if you measure the force distance profile in a low concentration monovalent ion solution, the force distance profile almost recovers to its original shape and range of repulsion. Now we ask so what is this structure? What is the structure of the collapsed brushes, as the surface force measurement only tells us the range of repulsion decreases? To first answer that, we tried neutron reflectivity. Now here, again, we started with a monovalent salt solution, and we see that we have a fairly diffuse PSS brush layer. When we added a very low concentration of trivalent ions, now the layer becomes slightly more compacted, but it's still fairly diffused and maintain a parabolic volume friction profile. However, if we add 0.1 millimolars etron ions into the solution, if you remember the surface force profiles, that's where we have a collapse of the brushes. We notice that the interference patterns are very different. We get regular interference fringes indicating we have a fairly compacted film. More importantly, when we look at the volume fraction profile, it doesn't show a parabolic profile anymore as a brush profile should have. Instead, we get this Gaussian or peak-like distribution. And this is really intriguing. Now, if you're familiar with the scattering data, reflectivity data of polymer brushes, now typically one would get such a volume fraction profile with a brush in the mushroom region. Now, because we have fairly dense PSS brush, we're definitely not in the brush mushroom region. We can also collapse the brush by using monovalent salt, high concentration monovalent salt. But there, we also we still get a parabolic volume fraction profile. So that's definitely something related to the trivalent salt. So we looked at the literature, which we've came across the studies on solvophobic effect on polyelectrolyte brushes. Now, about 2 decades ago, Julina actually published a couple of papers showing that for low grafting density polyelectrolyte brushes in poor solvent, they actually form this micellar structure, also called pinned micelles. Now, the principle is indeed fairly straightforward, as in poor solvent, the system wants to minimize the surface energy. And therefore, at a low grafting density, the smallest volume to surface area to volume ratio is a sphere, is a spherical shape. So that's why you have this pinned micelle-like structure. Now, this has been approved actually 2 years ago. We're using gold nanoparticles with polystyrene brush, which shows at low gravity density, one would have indeed pinned micelle-like structures. But when we increase the grafting density or increase the chain length of the polymers, the system will go to a uniform layer, because at high grafting density, there the smallest surface area is indeed a flat surface, because now you have enough polymers to cover the whole surface. And if we look at the surface, the grafting density of our system, we should have this uniform layer structure. But when we look at the reflectivity data, we are in this micellar-like structure. So we wonder what is the real structure we have to do that. To answer that, we did AFM measurements. We first measured it in monovalent salt solutions, monovalent salt solution, sodium nitrate, and showed that in both low and high concentrations of sodium nitrate, our profile is indeed fairly uniform. But when we started to add trivalent yttrium salts, we start to notice that inhomogeneities start to show up. And with the 1 millimolars yttrium, and even 0.1 millimolars yttrium, I should say, we measure those micellar-like structures with AFM. So the AFM measurement confirms we have micellar-like structures with the collapsed PSS brushes, although the grafting density is quite high, close to 0.1 chem per square nanometer. And we also did MD simulation in collaboration with the Pablo group at University of Chicago. Here we have a polyelectrolyte brush system with 64 chains, and the brush shows a uniform structure in the presence of monovalent salt. But when we start to add trivalent salts, we started to notice that the inhomogeneities start to grow, and with 1 times 10 to minus 3 trivalent salt in the solution, here I should say this concentration is a number of salt molecules per sigma cube, and sigma is the segment length in our of the polyelectrolyte chain in our simulation with 1 times 10 to the minus 3. Salt, where we have seen these micellar-like structures, which agrees with our AFM measurements really well. So we wonder if this is mainly due to sulfobic effect or due to a multivalent effect. And we did the surface force measurements again. Here we use isopropanol as the pore solvent because it can be mixed with water at any content. When we start to increase the concentration of IPA in the solution, we notice that the long-range repulsion between two PSS brushes completely disappear. The film actually completely collapsed to almost close to the dry film thickness. And during separation, we measure strong adhesion force between two PSS layer, which is typical when you do such measurements with polymer layers in poor solvent. Now, when we compare the force profiles between the poor solvent case and the trivalent salt case, they're very different, showing the multivalent effect is indeed very important. Also, again, when we did the AFM measurements in pore solvent, we seen that indeed the collapsed PSS brush surface has a very uniform morphology. Now, this again agrees with other people's study on gold nanoparticles really well because we have a fairly high grafting density, so we are indeed in this cold shell regime. Again, when we compare the AFM profile with in pore solvent with the AFM profile with trivalent salt, they're dramatically different. Again, emphasizes electrostatic cross-linking actually is very important now. To further elucidate the sulfophobic effect and the multivalent effect, we again did MD simulation. Here, by tuning the interaction strength between the segments of the polyelectrolyte chains, we can simulate the good solvent, the brush structure in good solvent and in poor solvent. In both cases, we see that the morphology is fairly, surface morphology profile is fairly uniform. Okay. But when we add trivalent salts, indeed, we can see that we see aggregation-like structures. Now, those structures are slightly different from the ones you see in the previous slides, because here we have actually a much higher grafting density, 4 times higher grafting density. So the structures are indeed larger. The effect is definitely not only limited to trivalent salts. So here, I actually just started to measure the effect of divalent salts, so those are very new results. We see that in the presence of divalent counterions, the brush also collapses, although you observe a much stronger ion-specific effects with different divalent ions. With barium, we kind of get the structure similar to trivalent salts. And with the magnesium, the surface profile is closer to a monovalent salt solution. Now, we believe this is related to the hydration structure of the 3 divalent ions, as well as the interaction strength of the 3 divalent ions. Now, with that, I would like to summarize my talk. We observed the lateral inhomogeneities of collapsed PSS brushes in the presence of trivalent ions. Those inhomogeneities are absent if you only use pool solvent. So based on that, we conclude that electrostatic bridging is very important in forming those inhomogeneous structures of a collapsed polystyrene softener brush. Thank you very much.
Thank you very much. Very interesting. Time for questions. Matthias?
Very nice work, of course. Congratulations. We have developed a very simple model based on the Alexander de Gennes brush. And then you have this exchange and done in equilibrium and so on. So it's a very simple model. And then you can see for this model the collapse, at least, or the early stage. Did you try to compare this with this model?
Do you mean that when you have this osmotic model, you can...
Right, yeah.
I actually looked at it. We actually get a very similar first-order transition-like structure change when we increase the trivalent salt. Now, I don't think in that model you can predict this micellar or inhomogeneous collapse
structure. But the micellar structure, this is the last stage. Mm-hmm. But I think you can, it's a smooth transition. Yes. And of course, it's not a phase transition, it's simply a smooth transition. The Alexander de Gennes brush type approach to describe most of it. Mm-hmm. Of course, it should not describe the pinned micelles.
Nice talk. I'm focusing on the large, the repulsive interaction, large distances. I mean, you didn't talk about that much, but usually this repulsion scale. I mean, it's the decay, exponential decay should scale with the bilengths. If everything is okay, so I'm wondering here with added trivalent ions, if you see the same behavior. If the ionic strength that you set at the beginning scale with the repulsive interaction, or something else is going on?
Well, certainly, if you decrease this, is especially the electrostatic repulsion is especially very important when you have low ionic strength. Now, when you increase the ionic strength, then the bilength decreases. Then it becomes less significant. Another thing we noticed is that in this particular case, the bilength is close to the fluctuation of the fluctuation range of the polyelectrolyte chains. And therefore, we believe the two are convoluted and it's really difficult to separate some circumstances.
One last question, Bjorn.
Yes, very interesting. Of course, PSS is quite non-polar, quite hydrophobic, so will easily associate.
Yes.
Due to hydrophobic interactions, you don't need to change the electrostatics so much. Do you have an idea about aggregation number in this?
Actually, very good question. One reviewer asked exactly the same question. Based on the size of the micellars, our aggregation number is fairly large, in the order of 10,000. You can also do a simple geometric argument because we know the size of the micellars, and we know the area behind it, roughly around the sizes around 200 nanometer in diameter. So based on the graphene density, it's also the same order. So we roughly know the aggregation number, but we don't really know the accurate aggregation number. The thing we don't really know is whether this collapsed structure has water or not. Because this is something I'm not quite sure with, indeed.
You can try solubilization or something to check.
Right. Another thing we're trying to do is, as you said, PSS is really hydrophobic. We're trying to remove the benzene group, just use the PSBMA to see if this actually plays a
change the hydrophobicity. Yes,
right.
Well, thank you very much indeed.
Thanks.